Hi, this is Professor Howard. This is the first of a two-part series of lectures on synaptic plasticity. This is some of the molecular mechanisms that underlie learning and memory in the brain. So I feel it's a very important topic to discuss in this course. And what you're looking at here is a diagram of a hippocampus that's shown in an artistic format. So this is artist Greg Dunn showing some connections in the hippocampus. Hippocampus is really important in short-term memory, long-term memory storage, and connections with the cortex for memory reconsolidation. So I've really distilled this very complex topic into two lectures, and the first part is going to be all presynaptic mechanisms of synaptic plasticity. Please pay attention to these 5 major concepts to master. If you have a really good feel for these topics, you should be good for this, this material. And then take a look at the PERVs chapter that I provided on Blackboard. I think it's a really nice primer for both this and the postsynaptic mechanisms, which will be covered in this subsequent lecture. So I'll start with memory, sort of a global view of what memory is and how scientists think about memory. We'll then talk about presynaptic synaptic plasticity. This is the presynaptic element's ability to change in neurotransmitter levels that are released. We'll then talk about heterosynaptic presynaptic facilitation. This is the idea that a second synapse can make a synapse with a connection and then change how its output is going to work. So let's dive in. So where is a memory stored in the brain? This was a really big topic very early in the field, and Carl Lashley was a researcher who decided maybe if we lesion or make damage to different parts of the brain, we can isolate where memories are stored. What he found is that it was very hard to find a specific region that would disrupt long-term memories. What he had to do was lesion about 70% of the entire cortex, so all different areas in the brain, before animals would forget how to run in a maze. So this led to the idea that memory was stored diffusely across the brain. That is, different aspects of memories are stored in different areas, and until you disrupt that large network, you can't disrupt memory. Now his student, Donald Hebb, went on to propose a mechanism for how this occurred. And he suggested that maybe these different brain regions are interconnected when learning occurs, and those those connections between those areas are strengthened when learning is taking place. That idea of a change in synaptic strength, which underlies learning and memory, has been known as Hebbian plasticity. So you'll come across this quite often in papers. So where are we thinking memories are stored now? There's this idea that a collection of neurons called a memory engram is a network of cells that incorporates or stores and can recall memory. So let me explain how this works. Imagine you have an animal that's learning a fear conditioning task. So you're playing a tone and then giving it some sort of scary stimulus like a foot shock. What happens is the hippocampus is activated, the amygdala is activated, and when those two things are activated at the same time, some change in strength between these regions occurs. So you see here there's also connections with the prefrontal cortex. Prefrontal cortex is not active during this learning, so these cells aren't strengthened, which is why they're not green in this image. And then what happens when the animal's reminded of this scary cue? Any one of these cells or any one of these areas becomes activated, and then the entire network becomes activated. So that's sort of like a single portion of the memory activates a subunit of this entire memory circuit, and then the entire thing gets recalled during learning or during recall. So what's happening in the brain that underlies this change in connection? There are 2 types of molecular mechanisms: changes in the presynaptic output of a neuron, so that's what we're going to talk about in this lecture, and then the addition of new receptors, the change in functioning of receptors, or new synaptic connections postsynaptically. A lot of these things are guided by changes in gene expression in the brain. That's going to be the content of the subsequent lecture. Okay, so we can't start a lecture on synaptic plasticity without first defining the term. Synaptic plasticity, very simply put, is a change in synaptic strength that is reflecting some history of activity in that connection. So this was very early on was characterized by Bernardo Katz. This is the person that the second Journal Club paper was focused on. And what he found is that if you stimulated a presynaptic terminal that's making a connection with a postsynaptic cell, and then stimulated it again a second time very rapidly, so that's that short delay here, you would get what's called synaptic facilitation. So here's this experimental setup. He's stimulating this input to this motor neuron and then recording postsynaptic potentials in the motor neuron. A typical action potential in the presynaptic neuron would result in a PSP of amplitude 1 millivolt. But then if he shocked the input a second time in quick succession, then you would see this second PSP that is amplitude of 2 millivolts, which is twice the size of the initial one. So something's happening and the signal is becoming amplified based on this short duration stimulation. So the first pulse gives you this, second pulse gives you 2 times this. Now what happens if you separate these two stimulation events by a long distance or a long delay? This results in synaptic depression. So here we have the PSP of amplitude 1 millivolt, and then we wait 200 milliseconds, give a second shock, suddenly the PSP has gone down in strength. So we're going to dive into some of the mechanisms that underlie these two ideas. And again, is this presynaptic or postsynaptic? And the short answer, and what we understand now, is it's both. There's changes both presynaptically and postsynaptically. Okay, one other thing I want to say about the term synaptic facilitation. This is a broad and general term that means an enhancement of neurotransmitter release or an enhancement of the strength between connections, but it also is a very specific term, and so I'm going to use synaptic facilitation to kind of mean a strengthening in general, but then also a very precise mechanism. So I'll try to draw a distinction so that's pretty clear. Okay, we're going to start with synaptic facilitation, and we're going to talk about 3 mechanisms that facilitate or enhance the strength of release in a presynaptic terminal. The 3 mechanisms are going to take place within the same synapse at which the change in strength will be monitored. That's true of synaptic depression as well. We're then going to talk about another type of plasticity that can occur where one synapse makes a connection to a presynaptic terminal and can alter its strength. Okay, so now, generally speaking, synapses can facilitate. The first mechanism I want to talk about is called presynaptic facilitation. This is a very precise definition, a precise mechanism. I hope that's not too confusing, but it's simply the way that it is. So what happens in presynaptic facilitation, just like what Bernardo Katz showed, you drive 2 action potentials in a presynaptic terminal. The first PSP is of this amplitude, which is about 2 millivolts or so, and then what happens when the second one arrives really quickly is all of a sudden it gets bigger. So what's underlying this? We know that neurotransmission is dependent on calcium levels, and so when you have an action potential arriving in a presynaptic terminal, calcium is going to come rushing in. This is going to prime the vesicle fusion machinery, so everything's going to be lined up and ready to release. But then, before that calcium can get cleared out of the presynaptic terminal, the second action potential arrives, and then an even higher change in calcium is going to occur in the presynaptic terminal. So at any given time when you have an action potential arriving, you have influx of calcium, and then it's going to be transported back out based on the calcium transporter. But this takes a certain amount of time, and if the second action potential can arrive before that takes place, you have facilitation. Here's the timeline showing a change in synaptic strength. Imagine at time 0 we have the first action potential arriving, and then at any time along this timeline the second action potential is going to occur. So if this happens really quickly, within let's say 100 milliseconds or so, then you're going to have a large increase in the second size of the PSP, and that's due to more neurotransmitters, vesicles binding the membrane, more neurotransmitter being released into the synapse on the second action potential. So another way to say this is synaptic facilitation is an increase in calcium above the initial increase where you get just a normal change in calcium based on an action potential. It also is an increase in the probability of neurotransmitter vesicles binding and neurotransmitter release, or P, based on Bernardo Katz's paper. M is equal to P times N. So again, this is a very fast mechanism. This is the fastest of the 3 types of facilitation we're going to talk about. It's gone by 400 milliseconds. Okay. The second mechanism that can drive, generally speaking, facilitation at a synapse is augmentation. This second mechanism is also dependent on calcium, but the molecular machinery that's at play is a little different and the time scale is different. So, because it's dependent on calcium, this is also an increase in probability of neurotransmitter release. If you look at the presynaptic terminal, this is a kind of a busy slide, so just focus right here. There is an exchanger. This is like a pump, except it's using a naturally existing gradient of sodium to exchange for calcium. So if you let a little bit of sodium in, that creates a driving force because sodium is aggregated outside of the cell. And you use that driving force to then pump out calcium against its concentration gradient, because there should be more calcium outside of the cell. This is a mechanism that is used to regulate how much calcium there is in the presynaptic terminal. So with a lot of action potentials arriving at a presynaptic terminal, the sodium gradient gets disrupted. When the sodium gradient gets disrupted, the driving force in sodium gets diminished, or it goes down, and that results in a slowing of calcium being extruded or effluxing from the cell. So that allows calcium levels to linger for a lot longer when this gradient gets disrupted. That's the first part of augmentation. The second part is within this presynaptic terminal, there are also calcium chelator systems. We've talked about chelators before, but what they do is they bind to calcium, they sequester it so that it can't do its action. When those systems get saturated, they can't respond to any further increases in calcium, and it takes a long time for those systems to recycle. So if we look at the timeline for augmentation, you see that it lasts for a lot longer than presynaptic facilitation. Here you see that by 4 seconds, augmentation has not gone down all the way. So the first action potential arrives, you get some big increase in calcium in this presynaptic terminal, and then even 4 seconds later, you give a second stimulus pulse, then you're going to still get an increase in that second PSP. Now, why do we get facilitation in one scenario and augmentation in another? The main story here is this requires more action potentials to arrive at the presynaptic terminal. So facilitation can happen just after 1 or 2 action potentials. I guess you would need 2 for facilitation to occur. So change in calcium based on the first action potential, and then that second build in calcium on top of that, right? But in augmentation, you might need 4 or 5 or 6 action potentials occurring pretty quickly, and that results in a long-term change in calcium levels presynaptically. You can ignore depression in this for a minute. We're going to talk about that at the end. Okay. Now the third mechanism that can drive facilitation and strength between a synapse is called post-tetanus potentiation. Don't let this term scare you. What a tetanus is a high-frequency series of stimulations. So if you're driving what's called a high-frequency stimulus train, or a lot of action potentials that occur in a very short amount of time, that's called a tetanus. Potentiation just means an increase in strength. So facilitation, augmentation, potentiation, they all kind of mean the same thing. But the mechanisms that underlie PTP, or post-tetanus potentiation, are entirely different. What happens here is after a lot of action potentials occur in this presynaptic terminal, then we get the second mechanism taking place. We have high levels of calcium, which is dependent on the mechanisms from facilitation and augmentation, but that can activate calmodulin. And when calmodulin gets activated, this is a calcium sensor, then you get activation of this protein called CAMK2. This is calcium calmodulin dependent kinase 2. Here he is in this figure. And what this does is it plays a really critical role in shuttling these reserve pool vesicles to the readily releasable pool. So this is not a change in P, which would be the probability of vesicle binding. This is a change in N, which is the number of vesicles that can be released at any given moment. PTP requires a lot of action potentials to generate this phenomenon, but it lasts for a really long time. This can last up to several minutes, and even longer than that. You see this never really goes back down to baseline. So this can drive a long-term change in synaptic strength. Okay, so those are the 3 mechanisms of facilitation at a synapse. So have an idea of the mechanisms and the timeline for facilitation, augmentation, and potentiation. Now, on the other hand, depression. This is the idea that a synapse can release less neurotransmitter on subsequent action potentials. One thing I want to say before I even dive into the mechanisms here, facilitation, augmentation, potentiation, and depression, they can all occur at the same time. Because they have distinct mechanisms, they can all be going on at the same time. So while these things are taking place, oftentimes you'll have 1 or 2 mechanisms occurring simultaneously. But let's focus on this idea of depression. Here we have an action potential. The second action potential has a PSP of a smaller amplitude postsynaptically. What's going on here? This is really straightforward, but you have a fixed number of readily releasable pool vesicles. As they bind, then you just have less vesicles that can bind. So with subsequent action potentials, the probability for depression goes up. So depression is therefore dependent on calcium because we need calcium to drive vesicle binding, and it's dependent on neurotransmitter release. So let's say we want to study depression and we take away calcium. What happens? Here you can see a change in PSP amplitude. We're starting with 100% here, and then after a long series of stimulation at high frequency, depression takes over, and you see that the amplitude of the PSPs postsynaptically goes way down, okay, before coming back. This is at a normal level of calcium. Now, if you remove calcium, you see that this depression goes away, and then augmentation, facilitation, or PTP, they all start to take place. So then those are the predominant mechanisms taking place, because again, these are all occurring simultaneously. So here we've sort of isolated the key component of depression, which is calcium levels. Okay. Depression has a fairly long-lasting timeline. You see that it can last for about 4 seconds, so it's sort of on the same time scale as augmentation. But the amount of depression is really dependent on how much stimulation is going in as well. Okay. So I want to summarize all these ideas in the next slide. Imagine, if you will, we have a long duration stimulation train where we have lots of subsequent pulses all arriving at a really high frequency. So here's our first pulse, here's our second pulse, third pulse, fourth pulse. This is happening, 4 pulses in every 10 milliseconds. So this is really, really high frequency. Here's the amplitude of the first PSP. So just focus on this for now, and you'll see this is amplitude of maybe 1 or 2 millivolts or so. Now the second PSP goes up. This is based on synaptic facilitation and augmentation, which isn't listed here but is also taking place in the background. So each pulse results in a slightly bigger, slightly higher PSP. Then at some point, the stimulation continues. Then you see that the PSP amplitude starts to go back down. That's because we're running out of neurotransmitter vesicles. So now in this blue box phase, we have depression becoming the primary mechanism that's taking over. So facilitation's still there. There's still high levels of calcium. There's still high levels of calcium from augmentation. But that also exacerbates the level of depression that can take place simultaneously. So then note that PTP is also taking place in the background here, but it takes a long time for this to occur. So the entire stimulus train goes to the end of the blue boxes here. We stop stimulating the presynaptic terminal, and then we give a test pulse later. And you see that this PSP is way bigger than the first one here. This is because the N, or the number of neurotransmitter vesicles, has gone up based on CAMK2 and the moving of reserve pool to readily releasable pool stores. Okay, so if you plotted the amplitude of these PSPs over time, you would get a chart that looks like this, where facilitation, augmentation take place early on. So again, this is amplitude X, this is amplitude 2X, so here you see a big increase initially. This entire stimulus train is about 100 milliseconds, so that goes out to about here, where you can see depression starting to take over. It also is taking over here. Now we're running out of vesicle storage pools, and therefore depression takes over. Potentiation takes a while to kick in, but you can see it's starting to take effect by about 200 milliseconds or so. Hey, George. My son's trying to get in. I'm going to persevere and see what happens. So what this chart also shows is a high frequency stimulus train of pulses occurring here, and then a second test pulse occurring later. And you see initially there is oh, sad baby. Initially there's depression because this depression is long lasting. But eventually, if you wait long enough for those readily releasable pools to recover, then you get PTP and a big increase, which can occur minutes later. That takes some time for those vesicles to be shuttled. Yeah. Okay. So again, the key take home here, these things are taking place simultaneously. So just think about the predominant mechanism at any given time point and what is happening inside the presynaptic terminal. Okay. So that is all changes in synaptic strength that's occurring at the same synapse where you're monitoring activity. I next want to move to a scenario where there's 3 synaptic terminals. There's a connection between pre and post synaptic elements, and then a third thing is coming in and modulating the way that those synapses work. Okay? This is called heterosynaptic facilitation. And again, this is where a third presynaptic element is going to manipulate what's happening in the presynaptic connection. So here we have, let's say, a sensory neuron, which is going to release glutamate. Here we have a motor neuron, which is going to play some role in muscle contraction. And then the third synapse here is a facilitatory neuron. This is often going to release a neuromodulator, which can include dopamine, serotonin, norepinephrine, a lot of these other types of neuromodulators. Okay, so we have an action potential that can arrive at this presynaptic terminal. That'll result in an action potential shown here. Whoops, let me go back. And then some PSP in the motor neuron of some amplitude, let's say 1 millivolts or so. Now, what happens when this nerve gets activated? So now we've got release of this neuromodulator. It's going to bind to a metabotropic receptor. That's going to result in increases in cyclic AMP. So this is a G sub S alpha subunit for a G protein. That's going to change the way potassium channels are functioning. Specifically, it can close potassium channels. That's going to result in greater excitability by moving away from the equilibrium potential for potassium. So functionally speaking, that results in a bigger and longer action potential and then a larger PSP postsynaptically. So here's this mechanism again. A modulator, it binds to a metabotropic receptor here, increases levels of cyclic AMP or DAG in IP3, if you remember that pathway as well. This can, don't worry about S-type here, but it can modulate K channels, potassium channels. These will close, action potential gets prolonged, and then increases in calcium get to be bigger because you're depolarized for longer. So this results in more transmitter release and a greater PSP postsynaptically. So again, it's not the activity here that's important, it's the activity here that's going to modulate what's going on presynaptically. That's why it's heterosynaptic and not homosynaptic. Okay, so some really seminal work was done studying this kind of mechanism as a process of long-term memory storage by a researcher named Eric Kandel. He won a Nobel Prize for this work, and now I want to introduce you to his model organism. Eric Kandel utilized aplesia as a model for studying memory because it has a very simple connection between a sensory neuron and a motor neuron, and then it has a facilitatory interneuron. So here's Eric Kandel talking about his work.
So we're looking at an aplysia stretched out. We're going to focus in on the gill and the siphon. We're going to apply an extremely weak stimulus so you can see the amplification with sensitization. Weak stimulus to the siphon, you see a modest withdrawal of the gill. Now we're going to frighten the animal, startle it, give a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is startled. The memory for this event is a function of number of training trials. So if you give a single tail shock, plotting here, change in reflex strength as a function of time, the enhancement of the reflex lasts minutes. But if we give the same stimulus 4 or 5 times, we get a memory that lasts a couple of days. So we have a short-term memory and a long-term memory, and we can now look at the difference between them.
So if you're not sure what you're looking at here, let me just walk through one part of this. This is the siphon, and if you stimulate the siphon, you get this gill, it will withdraw. So this is our sensory neuron, if you will. This is where the sensory neuron's projecting from. Here is the end result of the motor neuron, causing a gill withdrawal response. When he shocks the tail to startle the animal, that drives the neuromodulator to being released. So that's going to be the heterosynaptic facilitation that takes place. Let me go back and we'll dive into that mechanism a little bit more. I can't see my mouse. Weird. Okay. Let me hide this thing too. So this is how heterosynaptic facilitation works in aplesia. We have the siphon or the skin here. You stimulate this and you have an action potential in the presynaptic sensory neuron. This drives glutamate release onto a motor neuron, which results in withdrawal of the gill. Now, if you shock the tail and scare the animal, then that's going to result in serotonin release. Serotonin is going to bind to serotonin receptors on the sensory neuron, and that's going to change the way action potentials look in the presynaptic terminal. So, before you get this startling event, you have a normal action potential here and a normal amplitude PSP in the postsynaptic motor neuron, shown here in purple. After you get release of serotonin, then the action potentials get bigger and they get broader and slower. That results in a greater depolarization, more calcium influx, and then larger PSPs postsynaptically. So this mechanism is dependent on two pathways. The G sub Q alpha subunit of G proteins and the cyclic AMP pathway, which is G sub S. So here we have binding of serotonin, increases in DAG and cyclic AMP. That activates, respectively, protein kinase C, protein kinase A. This is going to result in broadening of the action potential by closing potassium channels and activating calcium channels. So it's going to change the way that those channels work, and that results in more calcium influx in the presynaptic terminal and more neurotransmitter release. So this is the definition of neuromodulation. Here we have this neuromodulator, which might sort of directly depolarize a cell a little bit, but primarily what it's going to do is change what happens when the cell does have an action potential. So if you're interested in Eric Kandel's work, he's very famous. He won a Nobel Prize for this work. I see him at every meeting I go to. He's always got the big red bow tie on, which is cool. Click on this link and you can see some of the students that he has trained in his lab. His students have gone on to become famous. They're working at UCSD and Harvard and all over the place. And someday you could end up in this sort of lineage. NeuroTree is an interesting way to see how researchers are connected to their mentors. So you can explore some of the people even in our department if you want. And you can think about the bigger picture when you're thinking about who you want to go and do your graduate work with if you decide to go to graduate school. So anyway, it's a cool website for that. Okay, so this explains sort of a short-term mechanism, but you might anticipate that cyclic AMP and DAG levels won't stay active for long. So something has to take place to allow this memory to occur over the period of days. That is the process known as consolidation, where you get a long-term change and long-term memory storage. So short term, we can explain this based on the mechanism on the previous slide, activation of protein kinase A and closure of potassium channels. But long term, this has to be some alternate mechanism because this won't last forever. So what happens here is we have a lot of serotonin release, a lot of serotonin binding its receptor. This activates protein kinase A in a really big way, which can lead to subsequent activation of this pathway. MAPK becomes activated. This is you don't have to know what this is, but it's just another kinase. It's going to activate this molecule, which you should know about. This is CREB. It's cyclic AMP response element binding protein. It's a mouthful. But the main point here is this is a transcription factor. So this is going to end up in the nucleus. It's going to bind to DNA, and it's going to change the way the genes are expressed. So you can end up with new synaptic connections or some long-term activation of protein kinase A based on changes in gene expression that occur over a very long period of time, including days or weeks. Okay? This is the idea that gene expression changes can support long-term memory through a process called long-term potentiation or long-term depression. So that's the last slide. If you have questions, please be sure to write those down and ask them during our in-class simultaneous sessions that we're going to have for question and answer sessions. And thank you again for your time. I hope everyone's well. And take care and be prepared for part 2.